This is the second of 4 videos looking at the kidney. In the last video, we focused in on the gross structure, and you can see an overview of that here. However, if you wish to have some more information on this, or you need to refresh your memory, then please go back to the video specific upon this. For today, we're going to be looking at ultrafiltration, and I did highlight that before this video, if you watch the last one, it would be useful to pause and go back to module 3 and have a look at the structure of blood vessels, in particular capillaries. So ultrafiltration is the process of filtering various different items out of the blood under pressure into the Bowman's capsule. So as you can see highlighted by the green box here, for today's session we are going to be looking at the renal corpuscle, and the renal corpuscle is the sort of culmination of the afferent and efferent arteriole, which have the glomerulus, the bundle of capillaries sitting between the afferent and efferent arteriole, and that is encapsulated by that Bowman's capsule, and collectively these structures are referred to as the renal corpuscle. Now, you may not be able to quite distinguish this from the image here, but the afferent arteriole is wider than the efferent arteriole, and therefore, because if you've got this narrowing, this is then going to lead to an increase in pressure as a result of this. So A-level, we actually need to go a little bit further than GCSE, and we now need to look at these structures and be able to articulate why at this Bowman's capsule this is described as having a 3-part filter. So that's what the sort of next part of this video is going to detail for you. So I'm going to detail each of the parts one by one, but I will give you a brief overview first. So we're seeing here at the moment the first part of that filter, which is at the glomerulus at the capillary bed, whereas we've just mentioned there is a build-up of pressure that has occurred due to the wider afferent arteriole and narrower efferent arteriole leaving the glomerulus. The second part of the filter is comprised of the basement membrane, and you can see that as what I've represented as the blue crosses. So this is sitting between the glomerulus and the Bowman's capsule. The third part of the filter comprises of the podocytes, and we will detail those later on, but this is a type of cell which lines the outside of the Bowman's capsule and has finger-like projections. So in this image here, I have zoomed into the capillary, and we see that in the capillary that you've got a number of components there at the moment. So you've got a range of different proteins. You've got some plasma proteins and then some smaller proteins there. You've got some glucose in the orange colour, some amino acids. There's representation of some red blood cells and also of some white blood cells there. Then if we draw our attention back to the structure of the capillary, you can see that you've got those endothelial cells, which make up the walls of the capillary, and you can see the small gaps known as fenestrations, and these are 1 micrometer wide. So at this stage we see a number of components of the blood being forced out. This includes water, urea, some hormones, glucose and amino acids. Fundamentally, anything that is small enough to fit through that 1 micrometer gap at this point can leave the blood. Now, as red blood cells and most of your plasma proteins are larger than 1 micrometer, these are going to stay in the capillary. So the second part to be aware of the filter is the basement membrane, and this basement membrane surrounds the capillary. It is made of type 4 collagen, and if you're trying to remember that, if you think about where a basement of a house is, then that is going to be at the floor of the building, and floor and 4 rhymes, so type 4 collagen. So as I said, this is surrounding the capillary. It forms sort of like a net or a mesh-like structure that surrounds that. And the holes within this are only 250 to 400 nanometers. So this means that anything that has left through the capillary then has to be small enough to then fit through the basement membrane, which has those smaller holes of 250 to 400 nanometers. In addition, being collagen, it has a slight negative charge, and that is then going to repel any molecules that also have a negative charge, such as your proteins. So what this means is that your part 2, your basement membrane, is going to differentiate between molecules in terms of both size and charge as the second layer of filter that your molecules passing from the capillary and from the bloodstream towards the Bowman's capsule have to be able to pass through. So the final and third part we need to talk about are the podocytes. So the podocytes are cells which line the outside of the Bowman's capsule, and they have these sort of finger-like projections that wrap around the capillaries, referred to here and labelled as pedicels. Now, as you can see, the pedicels, by wrapping around the capillaries, they create these filtration slits, and each of these slits is approximately 4 nanometers in size. So this is going to provide the third part of this filter, because now we have again differentiation in terms of size, as we have moved down to only 4 nanometers, through which any remaining filtrate from the blood is able to pass through to form that final filtrate that passes down into the Bowman's capsule. And that brings us to the end of ultrafiltration and the 3 parts of that filter. So just to remind you, the first part of that filter that we looked at was the capillary, and that is going to differentiate on size, with fenestrations being 1 micrometer wide. When we come to the second part, we're at the basement membrane, and that's going to differentiate on the basis of both charge and also on size, with gaps here of 250 to 400 nanometers. And then finally, we're at the podocytes, which line the Bowman's capsule, and these have again only a size of 4 nanometers and differentiate therefore upon size. So overall, molecules that can move through have to have a relative molecular weight of less than 69,000.